of myself, my colleagues at Vivpro, and our speaker, I welcome you to the next of the Vivpro Regulatory Brief webinar series. Our webinar today is entitled Accelerating Pediatric Drug Development, the Role of Quantitative Clinical Pharmacology. We are well past the days of looking up dosages of medications in dosing handbooks like Harriet Lane, which were largely empiric and based upon clinical practice. Pediatricians, other healthcare providers, and parents now have the expectation that medications that they use to treat the children in their care have robust research supporting the dosing, safety, and efficacy of children in all ages. But as we know, drug development in children where these important data would be collected is not easy. Healthy children have much more important things to do than participate in clinical trials. They go to school, they're learning to ride their bicycles, they're playing with their friends. Sick children, on the other hand, cannot easily participate, and only limited assessments can be made. They rely on their parents and carers for transportation, and there are limits to their ability to comply with their assessments. A couple of housekeeping items before we get started. Today's webinar is being recorded. We invite your questions and comments, and please find the chat box on your screen. If you're having technical difficulties, please reach out to Holly at vivpro.ai, and we appreciate your feedback as always by answering a quick poll at the end of the webinar. We are indeed fortunate to have the speaker today, Dr. Jing Liu, who is the Senior Director, Clinical Pharmacology at Pfizer Research and Development. She's an internationally recognized expert in this area, and she will be sharing her perspective. Since her graduation from The Ohio State University, Dr. Jing has spent her career at Pfizer, working on a wide variety of projects, including pregabalin for the treatment of seizures in children, and more recently, rimegepant and zavegepant for the treatment of migraines. She's the author of more than 130 publications, abstracts, and book chapters, and she has been invited to give presentations at numerous scientific society meetings, as well as a scientific exchange for sharing pediatric information with the PMDA in Japan and the NMPA in China. She currently serves on the board of the American Society of Clinical Pharmacology and Therapeutics and is the immediate past chair of the IQ Clinical Pharmacology Leading Group Pediatric Working Group. She's a founding member of the International Society of Pharmacometrics. Please welcome today Dr. Jing Liu.
Hello, everyone. Thank you, Joan, for the very nice introduction and for this opportunity. And thanks to all for joining. This topic is very near dear to my heart, and I'd like to share a few tips on accelerating pediatric drug development with quantitative clinical pharmacology. Oops. Yeah. So here's the outline of my presentation. First, I will introduce the evolving global regulations on pediatric drug development. Next, I will share my view on when, what, how to plan for an efficient pediatric program. Then I will give a few examples of quantitative clinical pharmacology applications to accelerate the pediatric program. Finally, I will wrap up with some concluding remarks. So pediatric drug development typically takes a very long time, resulting in significant delays between adult drug approval as well as a pediatric drug approval. It involves in the past, as Joan mentioned, I was involved in pediatric drug development for Lyrica. It lasted almost like 2 decades. Since the late 1990s, global regulations have been evolving to enhance pediatric drug development. So I will highlight the key regulations in the next few slides. So let's start with the ICH guidelines. The first ICH guideline on ICH E11 was released in 2000. It covers the issues when initiating pediatric study, a pediatric formulation, and the timing of pediatric studies, type of pediatric studies, pediatric age classification, as well as ethical issues. In year 2017, ICH E11 was revised by emphasizing the common scientific approach rather than the regional requirement. It also emphasized pediatric extrapolation as well as modeling and simulation to enhance the pediatric drug development efficiency. In 2022, a specific guideline on pediatric extrapolation was released, which is ICH E11A. The ICH E11A specifically emphasized on the approaches using in pediatric extrapolation. This particular topic was discussed in a previous webinar by Vivpro by Dr. Lisa Bollinger and Alison Dunn. I won't go into too much details on this ICH E11 extrapolation, but do like to highlight some involvement since then. The new ICH E11A emphasize the continuum of evaluating pediatric extrapolation. So instead of the FDA's decision tree we used in the past, it also provides a framework to access pediatric extrapolation, including addressing the similarity of disease and the response to treatment between reference and target pediatric populations, assessing the evidence knowledge gap, drug quality, data quality, and confidence to support pediatric extrapolation. And the pediatric extrapolation, either by exposure matching when there's a high confidence in similarity and with high quality data to support extrapolation, as in what is shown in the green zone, or by conducting dedicated pediatric efficacy and safety studies. If there is a significant difference, not much of similarity between pediatric and adults, or insufficient knowledge in similarities as shown in the red zone. And more often, as we will be in the orange to red zone by using innovative pediatric studies to address the knowledge gaps. For example, using biomarker or alternative response marker for bridging, Bayesian or single-arm trial designs to reduce pediatric sample size. I will share some of these examples later. The new ICH E11A also highlights that pediatric extrapolation can be applied to both efficacy and safety. So this slide shows the regulations in the US. There's a longer history of pediatric regulations in the US. The first pediatric use labeling rule was enacted in 1994. The Pediatric Drug Development Incentive legislation started in 1997 and became the Best Pharmaceutical for Children Act, or BPCA, in 2002. BPCA studies are voluntary and cover the entire drug moiety. The other legislation on Pediatric Research Equity Act, or PREA, became law in 2003. The PREA studies are mandatory and also indication specific. How many indications for a drug, and there will be PREA requirement for the number of pediatric studies. That's the case. So both BPCA and the PREA were reauthorized in the FDA Amendment Act in 2007 and made permanent through the FDA Safety and Innovation Act in 2012. So the same year, the Pediatric Study Plan, or PIP, requirement was introduced. Later on, the FDA also issued specific guidance on general considerations, clinical pharmacology considerations for pediatric studies, initially in 2014 and updated it in 2022. In 2019, FDA also provided guidance on general clinical pharmacology considerations for neonatal studies. You can see this very many FDA regulations when we considering pediatric drug development. And this slide shows the EMA pediatric regulation. Alongside the application of SAH guidelines, the pediatric regulation in the EU was enacted in 2007, mandating the submission of Pediatric Investigation Plan, or PIP. A specific guideline on the format and content of a PIP was subsequently released in 2008, and it was later revised in 2014. And in 2018, EMA also adopted a reflection paper on the use of extrapolation in the development of medicines for pediatric patients. I'd also like to introduce a couple other countries' regulations that's often involved in global pediatric drug development. The first one is on Japan. There's no specific mandating regulations in Japan. However, PMDA is a member of International Pediatric Cluster. So this cluster aims to harmonize the global regulations on pediatric drug development and the review on specific programs. And there are also some frameworks established in Japan to promote pediatric drug development, like the MHLW, etc. For the time considerations, I won't go into too much details here. China also primarily follows the ICH 11 guidelines. Over the past decades, as you can see, there's a whole slew of Chinese pediatric guidelines have been released in China. Although I don't expect you to read Chinese, but the latest NMPA guideline on quantitative methods in pediatric extrapolation from adult data includes many useful English references I highlighted here at the end, if you like to find some reference there. So by now you may have sensed, a common goal across the many different pediatric regulations, which is aiming to bring safe and effective medications to pediatric patients faster and more efficiently. However, the different requirements across countries can lead to also redundancy as well as inconsistency in pediatric program planning. So therefore, early consultation with relevant regulatory agencies is important. Despite the evolving pediatric regulations across the globe since 1990s, there's still an average delay from the initial adult drug approval to pediatric drug approval for about 8 to 9 years currently, based on a publication recently. So next, I will share some tips on when, what, and how to plan for an efficient pediatric program. So let's start it with when. And there are some regulations in the US and the EU regulations on the timing of PSP or PIP. In the US, it is recommended that the initial PIP should be submitted no later than 60 days after the end of phase 2 meeting, or as early as practicable, but before any phase 3 or combined phase 2 or 3 study in the absence of end of phase 2 meeting, or no later than 210 days before submitting a marketing application or supplement if there's no phase 3 or combined phase 2-3 studies. In the EU, it is recommended that the initial PIP should be submitted not later than upon completion of human PK studies. This means in adults, and cannot be after initiation of adult pivotal trials or confirmatory phase 3 trials. So as you may have noticed, these are the timelines provided from the regulatory agencies for the latest time to submit the pediatric plans, but not the timing for starting the initial pediatric program planning. So in my own opinion, the planning of a pediatric program should start as early as, before FIH start, the first in-human studies start, and during the initial planning of the clinical development plan. This is because there are many interdependent factors and the time-consuming activity needed to be considered in pediatric drug development, which I'll further elaborate on the next slide. So the key factors need to be considered in planning a pediatric study are listed on the left-hand side. We need to consider what's the medical need, including the prevalence of the disease, as well as the available treatment. What's the relevant pediatric age group? The age group could be very broad, but oftentimes it depends on the disease that need to be studied. So what's the expected pediatric dose? Younger or smaller kids typically need lower dose than the older pediatric and adult patients. What pediatric formulations are required? As you may have experienced, liquids or flexible oral dosage forms are preferred for neonates, infants, and younger children. Clinical safety and non-clinical toxicity should also be considered. For example, juvenile animal tox study may be needed to assess developmental toxicity for younger pediatric patients. And also the ethical issues. As John also mentioned, it's unethical to conduct pediatric studies in healthy participants, even for phase 1 PK studies. So unnecessary PK studies, unnecessary pediatric studies should be avoided whenever it's possible. The trial-related activities should also be minimized to reduce the burden to pediatric patients as well as their families. You may refer to the ICH 11 Revision 1 for more details on these particular topics. As you can see, the planning of a pediatric program could be more complex than an adult program. So quantitative clinical pharmacology plays a central role in addressing these complexities and identifying gaps using ICH 11A pediatric extrapolation and planning necessary pediatric studies, whether they are a PK, PKPD, efficacy, safety, real-world evidence, and or post-marketing requirement, and ultimately providing substantial evidence to support pediatric drug approval. The planning of a pediatric program is an iterative process and it requires collaborations across multiple functions within an organization and a collaboration with regulatory agencies. So I'd like to share some examples on how to plan for or accelerate pediatric drug development using quantitative clinical pharmacology next. So this first example is on pediatric extrapolation with a high level of confidence, which is the green zone as I showed earlier. In this case, exposure matching in pediatric patients in lieu of a dedicated efficacy study supported the approval of a pediatric indication. The medical needs here was complicated skin and soft tissue infections in pediatric patients over 2 months to under 18 years of age. It was agreed by the regulatory agencies that confidence is high in disease and treatment response similarity between pediatric and adult populations. Population PK modeling and simulation were used to determine the recommended pediatric dose of ceteris by matching steady-state PK exposure and the efficacy marker. As shown in the table to the right, steady-state Cmax and AUC in different pediatric age groups and adults are presented in columns 3 and 4, and the ratios of Cmax and AUC to adult values are shown in columns 5 and 6, which these values are all close to 1 at the proposed pediatric dose levels. The efficacy marker in this case is more than 35% of the time that the free plasma drug concentration exceeds the minimum inhibition concentration, or MIC, as shown in the table. As shown in the last two columns, the probabilities of achieving efficacy marker in pediatric patients are very comparable to those in adults. So this pediatric extrapolation approach using modeling a simulation led to the approval of a high-dose regimen of ceteris for pediatric patients with complicated skin and soft tissue infections, eliminating the need for a dedicated prospective pediatric efficacy study. The second example is also on pediatric extrapolation, but with a moderate to high confidence in the yellowish zone area, as shown on the right corner. So in this case, the application of quantitative clinical pharmacology reduced the sample size of pediatric studies and shortened the timelines on an IgG1 monoclonal antibody that intended for an inflammatory disease. The initial pediatric study design shown on the right, the upper corner of the right of the slide. It included a multi-arm PK/PD dose-ranging study with 80 participants, followed by a randomized placebo-controlled pediatric efficacy safety study with 120 participants. Later on, leveraging pediatric extrapolation guidance and the literature in similar drug classes, disease similarity and the response similarity between adult and pediatric patients 4 years of age and older were concluded. So as a result, a scaled down version of the pediatric plan, which is shown at the lower bottom right hand of the slide, requires only 12 to 16 participants in the first study, and combined with the pop PK and the receptor occupancy modeling and the simulation, this is a significant reduction from the previous 80% participant study. The modeling and simulation also further supported the pediatric dose selection for the second efficacy and the safety study by matching the exposure response in adults. In addition, placebo response similarity was also confirmed using literature data and the real-world data for both adults and the pediatric patients. So considering the recruitment challenge and the ethical concerns, a virtual control design was chosen instead of a placebo control for the pediatric efficacy and the safety study. These proposals and the rationales were endorsed by both the FDA and the EMA. So pediatric extrapolation and the modeling and simulation resulted in a more than 50% reduction in sample size and saved 12 to 18 months of time in this particular example. The third example is on pediatric PK and dose prediction, which is a key aspect of quantitative clinical pharmacology in pediatric drug development. Pediatric PK and dose prediction is complicated, requiring considerations of many factors, such as shown in the right of this slide, a broad range of age and body size, maturation of absorption and disposition pathways, especially for neonates, infants, and young children. the maturation of some pathways is still not well understood up to now. And the mechanism of drug disposition. Sometimes it may involve multiple pathways and or remain poorly understood, as well as the confidence in pediatric PK and dose prediction. The older the pediatric population, the similar and the better understood mechanism of drug disposition and the higher confidence in the prediction. Whereas the younger the pediatric population, especially neonates and infants, the more complicated pathways or less understood mechanism of drug disposition, the less confidence in the prediction. Safety window of a drug is another important factor. Wider safety window can accommodate greater uncertainty in pediatric PK and dose prediction, while narrower safety window requires a more precise PK prediction. All these factors can influence the decision on whether a dedicated pediatric PK study is needed, or if sparse PK sampling or only modeling and simulation is sufficient to inform pediatric dosing prediction. I'd also like to mention the pediatric PK prediction approaches, in which it often depends on the pediatric age groups. The traditional allometric scaling by body weight, which uses a power of 0.75 for clearance and a power of 1 for volume distribution, is more appropriate for prediction in older children. Allometric scaling plus maturation function can be applied to younger children, infants, and neonates, although the maturation function for some pathways have yet to be established. More recently, an age-dependent exponent method was developed using data from 73 drugs with different elimination pathways from neonates to adolescents. So in this approach, as shown in the table on the left, the power exponent is age-dependent. It's become bigger as the age become smaller. Using this approach, the prediction as shown in the figure on the right with the blue symbols, the prediction from the age-dependent exponent is pretty much comparable to the predictions using a PBPK model. I would actually like to suggest people to try out this approach and confirm, this utility in the future pediatric drug development. So coming back to PBPK prediction, it often relies on a more mechanistic drug disposition as well as the physiological parameters. Although in some cases, the drug disposition may not be well understood, and some of the physiological parameters for young infants, especially, or neonates, are not well established. It still have some limitations, but once well established, can be applied to all ages. The other aspect I want to highlight is on the prediction in adolescents. In the FDA's publication, it states that 94.5% of the drugs have identical dosing for both adolescents and adults. This implies that for drugs with a sufficient safety margin, adult dose could be applicable for adolescents without further adjustment, and adolescents could be included in adult drug development programs eventually. So leveraging this quantitative clinical pharmacology, we can provide accurate pediatric prediction to the dose and PK, and reduce unnecessary pediatric PK studies and or pediatric PK sampling. This next example is on the planning of age-appropriate pediatric formulations. As mentioned earlier, a wide range of pediatric ages may require different formulations, different dose strengths, and or delivery systems to facilitate drug administration. The figure on the right shows the potential exposure ratios between pediatric to adult by body weight, assuming the same adult dose is given, and also using the age-dependent exponent prediction. The exposure ratio remains within twofold, down to a body weight of 30 kilo. However, for younger pediatric patients with lower body weight, the exposure ratio could increase significantly. So this suggests that at least one or more pediatric dose strings would be necessary if the target population includes pediatric patients with body weight less than 30 kilo. This is just one example, just showing what could be the starting point when we're planning for pediatric dosing strings or formulations. So regarding formulations, a smaller pill size, coated or chewable or dispersible formulations may be desirable for children. Solution, suspension, or multiple particulate may be necessary for newborns, infants, or younger children. Developing these pediatric formulations is often time-consuming. So quantitative clinical pharmacology plays a key role in predicting potential pediatric dose range early in the development program, and leading the relative bioavailability, bioequivalent, and the food effect studies for this age-appropriate formulation development. Engaging with cross-functional teams, especially formulation scientists early in the process, is critical to determine the most adaptable and the minimum dose strings, thereby preventing delays in developing age-appropriate formulations. This next, before I go to the next example, I also like to mention a few considerations on pediatric dose selection, special considerations. The previous examples mostly probably applicable to small molecules. In some situations, the disease conditions can affect the drug elimination pathways and therefore impact the predictability of allometric scaling. For example, tofacitinib, which is a JAK inhibitor in inflammation disease treatment. The power exponent in pediatrics are actually 0.3 and 0.5 for clearance and volume distribution, respectively, much lower than the empirical values of 0.75 and 1. This is probably due to the cytokine impact on CYP enzyme activities. Another situation is renal hyperfiltration that has been identified in patients with sickle cell diseases using some real-world data analysis. Therefore, when predicting pediatric PK and dose for renally eliminated drugs, renal function must be taken into consideration in these disease patients. And for different drug modalities, such as therapeutic proteins, including fusion proteins, monoclonal antibodies, target-mediated drug disposition may impact pediatric PK and PD prediction. A difference in target levels, So difference in target levels between pediatrics and adults should be considered in pediatric PK and dose prediction for the monoclonal antibodies and the fusion proteins. Another factor is immunogenicity, which may also impact the PK and the PD. And differences in immunogenicity between pediatric and adults should also be considered in pediatric PK prediction. For the oligonucleotides, cell or gene therapies, I think we are still at the learning stage on pediatric PK and dose prediction, and information may evolve, and we will continue to learn on these special cases. And this next example is on utilization of novel data and modeling tools to enhance pediatric program efficiency. This work is published by a cross-sector collaboration from the Cooperative International Neuromuscular Research Group and the Duchenne Regulatory Science Consortium. In this case, data from randomized controlled trials and the real-world data were used to build a disease progression model for Duchenne muscular dystrophy. A model-based clinical trial simulation platform was then developed to optimize clinical trial designs for DMD. This platform can be utilized to perform trial simulations to optimize patient inclusion-exclusion criteria, endpoint selection, sample size optimization, treatment enrichment, and other clinical trial parameters, as well as dose selection by including drug effect models. This last example is actually a successful outcome or case study for the innovative study design in rare disease pediatric population using external control, natural history, and disease progression modeling. This is a snapshot of Brineura, a US label. The efficacy of Brineura was based on a non-randomized, single-arm pediatric clinical study. Brineura treated pediatric patients were compared to untreated patients from an external natural history cohort using disease progression assessment. And the result of the success of the comparison actually are successfully incorporated in this drug label as highlighted on the slide. So in summary, quantitative clinical pharmacology plays a central role in accelerating pediatric drug development. Early planning and the cross-functional and cross-stakeholder collaborations are critical. Modeling and simulation can advance quantitative assessment of pediatric extrapolation, novel data and technology, pediatric dose prediction, and innovative pediatric clinical trial designs. Early engagement with global regulatory agencies, more harmonized regulatory requirements, and streamlined reviews can further accelerate global pediatric trials and ultimately achieve the convergence of more efficient pediatric drug development. So with that, I'd like to conclude my presentation. And thank you very much again for the opportunity. And thank you all for your attention. Happy to answer any questions.
Thank you very much. What an informative lecture you've given us with, the additional things to read and catch up on later. Several questions have come in. The first is, if you have the same guidance, but one is a draft and more recent, and one is previous edition, but it's finalized, what do you do?
Mm-hmm. Oh, that's a great question. Yeah, as I mentioned, in the different regulations, many guidance, have been updated throughout the time. I would personally recommend look at the most recent version of the guidance that's reflecting, the most up-to-date knowledge, as well as perhaps, correction of some of the outdated knowledge. Mm-hmm.
And certainly it's a point you could perhaps clarify in communication with whichever agency you're working
Mm-hmm.
with.
Yeah. Mm-hmm.
Another question, and this has to do with the infectious disease example that you provided. How did you calculate the probability of attainment for the pediatrics in the first place? Was it based on the adult data? Also, if you want, there's several parts to the question. So if you get to the one there, this is it. Yes. So the pediatric target attainment, was that an assumption, or did you actually have additional data, or were there additional data?
Yeah. So in this case, actually, the PD marker or a target value of 35% of time that the free drug concentration is above MIC that was established in adult population. So that's the target when we predict or select the pediatric dose. That's the target we were matching to achieve that same target as in adults, because we established, the disease similarity and a response similarity between pediatric and adults.
Thank you for the clarification. How did you predict the dosing for the children less than 2 years of age using population PK?
That's another great question. Maybe I'll move to that particular slide, which showing the different approaches. Definitely, it's not the traditional empirical allometric scaling is not recommended for pediatrics 2 years old or younger, because it doesn't take into the maturation and the ontogeny on the different pathways. If you can find the maturation function in the literature using POP-PK, including allometric scaling as well as maturation function, included the reference below. You can do population PK based prediction. As also mentioned earlier, this newly published age-dependent exponent methods you can try, because the method was developed based on data from neonates all the way to adolescents, and seems like also the prediction is very similar to the prediction from PBPK models. That's a couple approaches we can use POP-PK to predict PK and dose for very young pediatric patients.
Thank you. Now, you mentioned immunogenicity, and we understand that immunogenicity can affect pharmacokinetics. Are you familiar with where it also affects dosing?
Yes, I'm aware that the immunogenicity may affect dosing, particularly because it may affect the efficacy of the drug. If it's a neutralizing antibody to therapeutic proteins, not only the PK may be impacted, but the efficacy could also be impacted. Therefore, there'll be a necessary dose adjustment, or there could be a loss of efficacy over time with the development of anti-drug antibody in this case.
Thank you again. The questions keep rolling in.
That's great. Yes.
Excellent presentation. I absolutely agree. You mentioned the use of POP-PK for populations 2 to 24 months, but in general, we use PBPK approaches for those less than 2 years. Is it based on therapeutic classification, or how would you choose POP-PK versus PBPK?
Yeah, so that's another great question. So in terms of using PBPK, as mentioned earlier, we need to understand the ADME properties of the drug very well, so that we can not only accurately predict pediatric exposure. First, we need to validate the PBPK model, which should have a good prediction even for the adult data. Only after we have high confidence in prediction of the adult data, then we can utilize for prediction of a pediatric PK and dose. In some cases, the ontogeny of the certain pathways, either CYP enzyme or certain, especially certain transporters, are still not well established. So in that case, when we use PBPK, there still will be limitations to predict PK in pediatrics 2 years and younger in that case. However, this age-dependent exponent approach is, I would call it still quote empirical, but it was established across many drugs, 73 drugs, as I mentioned in the literature, and across a wide range of age from neonates to adolescents, and included many different mechanisms of drug disposition. I would like to suggest give a try if you have limitations in the PBPK prediction per se.
Thank you again. Many drugs in children are dosed by body surface area. So if your exposure matching from adults to extrapolate pediatrics is used, but the adult PK model only has weight included, what do you do?
Oh, that's another great question. So oftentimes we know like body weight and the body surface area are highly correlated. Yeah, so it's hard sometimes to differentiate, the body weight versus the body surface area. In terms of modeling, we could include the body weight in the modeling per se. We will get, the absolute amount of drug each subject administered, and also the actual body weight from that subject. We can still evaluate that relationship, but there will need to be some post-analysis process where we could convert to, the body weight to, body surface area based modeling. So that's, I guess, not straightforward, but it can be done.
Exactly. So to translate it to common units, perform the modeling activity, and then go back and give each group their appropriate information for dosing.
Yes.
Now, there's one question on slide number 18. The redesign of the pediatric study was this redesign based on pediatric data? And the person also would like to know, could you further elaborate on what made the team remove the age stratification in the PKPD study?
Yeah. So the original, again, great question. The original study was designed with also the focus identifying different doses. As you can see, not just only one dose, the low dose level, there was a high dose level, primarily based on some statistical considerations to, give appropriate sample size. With the confirmation of, the disease similarity and response similarity using similar, drug classes, the design of the new design for that study is actually based on PKD exposure response. In that case, not necessarily we had to size the study based on statistical considerations rather than characterizing the PK and occupancy relationship in this case.
Thank you. That was great. One final question. For body surface area based dosing, is there an exponent that's typically used for allometric scaling?
Great question. The answer is yes, and it has been older approach. There are some literature, but I cannot remember the exact value for body surface area based scaling. But I do want to highlight, if, for example, the adult dosing is already a body surface area normalized, and if the intended dosing in pediatric population is also body surface area based dosing, in that case, the similar or potentially the same body surface area normalized dose could be applicable to a pediatric, no matter how low the body weight. Actually, I had a backup slide. I thought I would not use it. I don't know if Holly, you could put that backup slide on the screen so we can show to the audience had like a hypothetical simulation, even though it's not particular body surface area per se. I use a body weight normalized dosing, but it would be similar to body surface area. In that case, it could be very, maybe go all the way to the end. There's a figure. Yes. So this is the figure. Use a body weight normalized dosing across different body weights. You can see the exposure ratios from pediatric to adults across the body weight is pretty much within the fold of 1.5 in this case. So unless there are special concerns on the safety and the tolerability, if the drug has wide enough safety margin, the same amount of surface area, a dose, body weight normalized dose could be applicable to pediatric patients across a broad range of body weight. Thank you, Holly.
Well, thank you very much. Is there any concluding bottom line you would like to leave the audience with?
The bottom line, I think, is this, as I mentioned in the concluding remarks. Definitely, I like to suggest let's plan the pediatric program early. We don't need to figure out all the details, at the first in human stage, but still, if we could consider the key components and make sure we have timelines, identified when we need to, start dive into in more details in those timelines, those are important. And also engagement with regulatory agencies sooner, earlier, and hopefully get more consistent feedbacks from different regulatory agencies that will help, the more streamlined pediatric program. And the last, is my main message for this presentation or for this webinar is the utilization of quantitative clinical pharmacology to integrate all the information, so analyzing all the information we have and identify the gaps we have and design a more specific, efficient pediatric program.
Thank you again for an absolutely wonderful presentation.